Guys. I hope you guys can see the slides. You can hear me properly. Um, I do not know what is the way of communication, but is there any way? Please let me know if you cannot hear me. If there is any problem, then we'll try to address that. Okay. So myself, Dr. Praveen Bansali. I'm a professor in Department of Chemistry at Alliance University, and we guys are really excited to see you here for the BTech program. Um, we're gonna start it soon. I hope same stories there on the other side. You guys might be excited to join here at Alliance University for the program, which you are looking forward to it. Today we're going to talk about applications of engineering field at the interface of first one, engineering and chemistry, and second one, engineering and biology field. So this is many times we are not clear with this one that whether applications are involved there toward the chemistry side or of the chemistry toward the engineering side, or of engineering side toward the biology side, or of the biology toward the engineering side. And many times that is the reason we miss the opportunity to make our career in those particular field. Let me tell you one quick story and then we'll keep on moving. Those who work on the interface, when I'm saying interface, mean those two disciplines. For example, engineering and chemistry, these are the two disciplines, separate disciplines, we have it. And those who make career there at the interface of any two fields, then they are in real demand, because it's really difficult to find a person who is expert in the field, both the field, engineering as well as chemistry, or engineering and biology. So somebody who comes with those expertise, then that person is in real demand. So that we have to understand, and that is the kind of things I have learned it. I have done my PhD. You can see that from United States of America. And when I was doing my PhD, when a friend of mine, he was doing MD. MD is equivalent to MBBS, and PhD, MD, PhD he enrolled for, and PhD was in medicinal chemistry. Okay, although first word is medicine, but the second word is chemistry, and we used to do very heavy chemistry. Generally, you know very well, those who are going for MBBS program, they are good in biology but not in the chemistry. Generally, that is the trend. We have it in India, and it's really difficult to find a program in India which can offer you a curriculum which will serve the purpose of working across the field, okay, or the interface of two subjects. So that's really difficult, but that is one story. And then I moved to Johns Hopkins University, which is one of the premier research organization in not only in the United States but worldwide, that is famous for research and hospital. Okay, so many of the celebrities, when they are suffering from terminal diseases, they go to the Johns Hopkins Hospital. The beauty of that hospital is that hospital is on one side of the road, and the other side there are research institutes are there. And why I'm mentioning that, I'm again mentioning that somebody I met there, my ultimate advisor, he was again MD, and he had a PhD degree in organic chemistry. So he used to work on the both the sides of the road. That is, he used to work in hospital. At the same time, he used to work um, in research, toward the research side, in the chemistry side. He was equally successful. He was good radiologist. He was a radiologist. And at the same time, when I moved away from him, he at least had 10 grants of $1 million. So having that kind of a success story behind you, that is really kind of like an achievement in itself. At least 150 people might be working for him when I moved in 2016 to India. Okay, so try to understand. So if you somewhere can make your career at an interface, then you are already set for your life. And try to understand, we that is the thing. When we are going for some education, we want to find a livelihood, right? Or somewhere we want to settle down in one field, so that there is no challenge for our livelihood. Okay, so let's begin our talk. First of all, we'll deal with application of engineering field at the interface of engineering and chemistry. The first word is polarimeter, and then purposefully I put this one, polarimeter. Polarimeter, you might have learned it in case of chemistry, this word. Polarimeter is instrument which helps you to define the optical activity. When chiral compounds are there, you might have heard this in case of organic chemistry, to be specific, in case of those halogen containing compounds. Okay. So when you have chiral compounds, those are optically active. You might have heard those dextrorotatory, levorotatory, which rotate the plane polarized light in a clockwise direction, anti-clockwise direction. So polarimetry is an instrument which can help you to define the optical activity of compound. When the compound is optically active, so basically this instrument looks like this, which is shown on the slide. So this part somewhere you put the sample there, and this is the eyepiece. If you can see there, the cursor, I'm moving it there. So this is the eyepiece where you put your eye and then try to locate it. Okay, and then how that looks basically, when you see through this aperture, through this eyepiece there, then what you look, you see a circle there. Half of the circle is illuminated, that is bright in color, and half of that is in dark in color. Okay, so somewhere that aperture has one line there as a diameter. Okay, so that line you have to adjust it across the bright and the dark field. So you have to do it this manually. During my PhD, I have done my PhD at University of Toledo, United States. So during my PhD, I have heavily used this instrument, and I know that the kind of a pain you have it there. Okay, so the pain, what you have to adjust that eyepiece, that this can rotate. This eyepiece, you can rotate it in a clockwise or anti-clockwise direction, and you have to align that line there, the diameter there, across dark, and half of that is dark, half of that is bright. You have to align that line there. Okay, so that is cumbersome. And why it is cumbersome? First thing, that what's your the dark region and bright region I'm mentioning, that is not very well defined. Like sometime if it is not properly defined, then it put lots of strain on your eyes, one thing. And then you have to adjust it properly, otherwise your reading will be wrong. Okay, so basically it differs from person to person where that alignment happens there. Okay, so basically manual error can happen there. It puts lots of strain on your eyes, and then results will be wrong. So these are the couple of disadvantages with this instrument. But the beauty later on, what happened toward the end of my PhD, my department purchased new instrument. And the beauty of that instrument, I have to put the sample, and then I don't have to look through that aperture to the eyepiece. I just have to go to a PC. I just have to click there. Once I click there, then right away the readings are there. So basically there is a communication between this computer and what's your that polarimeter instrument. We have it now. See, the life is really easy. First thing, there is no strain on eyes. Second thing, readings are right, and no manual error. One person is saying something else, second person is saying something else. That will not happen because we have given that command to the PC. And again, try to understand two things there. I'm going to mention it. Polarimeter, this is the instrument. Who is going to build it? As an engineer, you are going to build it. I'm a chemist. I don't understand anything about that instrument. Only thing is I know application of that instrument. Okay, so who is going to build that instrument? You guys going to build that instrument. So again, try to understand. We have immaculate kind of applications of engineering field across any field, not only chemistry, not only biology, not only pharmacy, not only physics. You take any subject, lots of applications are there. Unfortunately, we do not know. That is the reason we do not make our career. So if you guys are equally excited about engineering and chemistry, or engineering and biology, or engineering and physics, then this is the field for you guys. You should try to make your career across the field. Are you following it? At the interface of these two subjects, that would be really great. Once you are settled down there, then there is no challenge for your job. So either job, or even you can build your own instrument, or you can build the communication program. Now, what is the communication happening between polarimeter and the computer? Basically, you need to have that language, right? You have to build that program. And who is going to build that program? You guys, as a software engineer, you have to develop that program, right? So that's, try to understand that once you know those two things, then definitely you can build the program. And that is what you have to look for such kind of things, where what entices you, what attracts you. Apart from engineering, if something else attracts you, definitely you should go for it, and you should try to find it out maximum about those two subjects, and then try to find a niche area where you can make career of yours. Try to understand that. Now, next one, we are going to talk about NMR spectroscopy. A quick thing about this instrument. This one, it looks like this. This is NMR instrument there. So let me quickly mention that this NMR, when I was doing master's program in India, we had only one NMR machine. I had to synthesize my compound in laboratory, and then I wanted to use this NMR machine to decode what I have synthesized it. Okay, so I need NMR machine, and then when I was trying to find it out, there was only one instrument in that region, and I was doing my master's program in one of the kind of a educational hub, which is known for educational hub, cultural hub. That is Pune. Pune is in Maharashtra. This is well-known city, and despite of that, there was only one NMR machine accessible to the public. That was with the University of Pune, and that was only 150 megahertz. Okay, 150 megahertz. And the wait time for that instrument was 1.5 months. That means if I submit sample today, then I have to wait for the result for at least 1.5 months. So that kind of thing. Now let me tell you, when I moved to University of Toledo, we had in my own department, in my own basement, we had 3 NMR machines. If you can recall, I told you the one with University of Pune was 150 megahertz. Now at University of Toledo, we had 400 megahertz, 500 megahertz, and 600 megahertz. Okay, so that means as the number is going, of course, the resolution is going up, and at the same time, the cost of that instrument is going up. Okay, now so I was teasing one day with my advisor, because he did his PhD in 1976. Maybe most of you might not be born at that time. You guys are sure, right? You guys were not there at that time. 1976. Anyone was there? Hopefully no one was there in 1976. So he did his PhD in 1976, and I was teasing him that maybe at that time when you were doing PhD, yeah, PhD was very easy, or the chemistry was very easy, because you might not have NMR. He said, no, NMR was there. That was not an issue. But the issue was that what he told me. So basically, in case of NMR, what you get a graph. We are going to look at it in the end. How the NMR looks like, the graph, I mean to say. So that graph, they used to plot it on a graph paper. Okay, so basically you get different kinds of peaks, and you have to measure area under each peak. Okay, so they used to plot it on the graph, and then manually they have to count those number of boxes. You can imagine those graph paper small boxes, you have to count those. Sometime it is half, sometime it is covering only 1/4 or 1/3 or something like that, and somebody will say, no, this is more, this is less, you should count it, you should not count it, something like that. And it's very tedious job, one thing. And manually how to do it, so it will it's very time consuming, right? And then errors will be there. Manual errors will be there. Somebody will say, no, this should be counted, this should not be counted, something like that. Argument can be there. So it's very tedious. But when I was doing my PhD, I don't have to do this one. The beauty of this one, this NMR machine was, I put the sample there, I go to the PC, that is computer. I just give the command in, depending on which kind of experiment I want to run. Generally 10 minutes, that is done. Sometimes we used to do it overnight also. That is a different story. But then all those peaks, I used to see it on the piece of PC. I just give the command, then it will give me the area under each peak. Okay. And if you want to see it, you can see it. If you want to print it, you can just connect it to the printer. I mean, printer used to be there all the time, and just you have to give the command, print is ready for you. So try to understand how easy you are making the life of chemists. This is a challenge for you guys, that you have to find a problem around you. Society is facing lots of problems. For example, here we are considering the chemistry society here. Okay, so we people suffer lots of such kind of problem. I just defined that problem. That what is the problem? That graph was coming on a graph paper, and then manually we have to count the area under each peak. That is very tedious job. Manual errors will be there, time consuming. But on PC, just it takes only 1 minute to give that command. Everything is settled for you. The business is settled for you. Are you following it? So this NMR machine, and again, let me tell you one more thing. When I was about to finish my PhD program at University of Toledo around 2010, they purchased another instrument which was 800 megahertz. If you can recall, we have mentioned that earlier they had 400, 500, and 600 megahertz. This time they purchased 800 megahertz, and the cost of the instrument at that time in 2010 2011, at that time the cost of the instrument was $1 million. I'm talking in millions, and I'm talking in dollars, US dollars. So you can imagine almost 80 rupees exchange rate, right? $1 is equivalent to almost 80 rupees. And then I'm talking in millions. Millions means 10 raised to 6, right? Those who are aware of it. So you can calculate that. That will cost you a few crores. I think almost 10 crores, 8 to 10 crores to purchase this instrument. If the instrument is that costly, then you have to understand that even if you understand the what is called as construction and working, how to take it apart and how to put it back, even if you understand that, your life is set. You will be wondering that lots of institute has this NMR machine here in Bangalore. Not only I'm talking about universities, even pharmaceutical industries are there, chemical industries are there in Bangalore. So lots of those people have the NMR machine, and they need expert person who can take it apart, or those who understand it. That if something is going wrong with the instrument, how to fix that instrument, basically maintain that instrument. Are you following it? Maintenance engineer, they need it. So basically you need one engineer. And second thing, try to understand same thing. All machines can be attached to the computer, right? So just now we talked about how the life becomes easy. So if you can develop that program which can communicate between the machine and the computer, then life is set. And then somewhere you are making that process easier for the chemist, or whoever is suffering from the problem. So try to understand. So basically, even if you understand that construction and working, forget about, we talked about $1 million. You will be wondering that how we'll bring the money to if somebody wants to do the business, or somebody wants to set up the industry. That can happen later on. But even if you understand construction and working of that, you can become consultant. Let's say you are working with NMR machine for 10 years, and then down the line, somewhere I know that XYZ person who was my student, and he knows nitty gritties of that NMR machine. Then definitely if I want to purchase that tomorrow for university, then I'm going to contact that person. So basically will act as a consultant for me or for the university. Try to understand that. So that will bring laurel, that will bring money, that will bring the job for you. So basically what you need, you have to become expert in that area. So basically you need a good driver to run that machine, right? As I have shown this in the picture, that the driver should be expert. That driver should know how to drive onto the road. Otherwise, that can create a mess, you know very well. So we don't want such kind of scenario, which I have shown there, that all the monkeys are there and then running the car. So we have the same scenario where cell phone kills us, right? Everyone is busy with the cell phone. So that is the story. So even if you give the cell phone to the monkeys, I guess they will deal with it in the same fashion. So you have to decide you want to become expert in one area. So this is the time, I guess, to develop that expertise there. So basically we'll settle down with this driver who looks very cute, nice, right? Who has a tie, goggle there, very smart driver, right? So we'll settle down with this driver. I wish we can interact, but unfortunately it's not there. So in the next instrument, we are going to talk about high performance liquid chromatography. The short form of that is HPLC. Earlier it used to be called as high pressure, because how that I just quickly will define that how that works actually. This is used for separation purpose, okay, or for purification purpose. So basically you have a pump. The pump will pump. If you can see that I have purposefully taken, nowadays you have very sophisticated instrument, purposefully taken the picture which shows you kind of small things, nitty gritties, that how that looks. So if you see there lots of tubings coming and going, lots of things are happening there. So there is through these small tubings, with pressure you are pumping the solvents. Again, we are not going to define in detail solvent. Water is one of the solvent. So basically solvents, any liquid which is used in case of chemistry, that is pumped through these small tubings and through these small pipes there. That small pipe is filled with dirt. I'm talking dirt, but basically they fill the silica, but just for our understanding, to make it easier, that is filled with the dirt, and we are, that means if it is full with dirt, then we have to pressurize that solvent there, right? I hope you understand. And basically that helps to for separation purpose. So this instrument looks like this. Again, life will become easier sometime, because we use one solvent, two solvent, three solvent, we have to mix it, and then we have to give the command that okay, if I want solvent at 1 ml per minute, or from the another tank I need 2 ml per minute, from another tank I need 3 ml per minute, I should be mixed in that fashion. That program should run for 25 minutes, 10 minutes, 15 minutes. You can give all those commands, and all these commands, where are you giving? Basically HPLC is attached to the PC, that is computer, and then all the commands can be handled with the help of computer. So that is there. So these are the columns where that dirt is filled in and the compound is passed through, and then separation occurs there, or purification takes place. This is a sophisticated instrument. Lots of now kind of fancy things are associated with this one. Sometime you can give the command, and overnight you can run it. So overnight, without monitoring it. So just give command in the morning, the samples will be ready for you, analyzed and purified by the HPLC. So you have automated system there, all those things, lots of fancy things have come with this HPLC. And then again, try to understand this is engineering piece. You have to build it. Second thing, there should be communication between HPLC and that computer there. So again, you have to develop that language. So that who is going to do it? You guys are going to do it. And we, as a chemist, we do not know. We just know the application side of it. Then before we go there, HPLC cost you almost around. It comes in different packages, but minimum cost will be 25 lakhs. Okay. So again, these instruments are very costly. This GCMS, gas chromatography mass spectroscopy, I think the prices starts from 50, 60 lakhs and then go onwards to 3, 4 crores and to higher than that as well. Again, this is a piece of instrument. We are going to not go in detail about those things. The only thing is I want to introduce you that yes, what's your pieces of instrument we have it in chemistry lab or physics lab or any other lab, those are built by engineers, one thing. And then second thing, um, if some problems are there associated with that instrument, and who is going to find the solution? You guys going to find the solution. And the problem, I just defined it, that okay, that it is for plotting onto the graph, or it is putting strain on the eyes. So basically you try to attach all these pieces of instrument to the computer, so that life becomes easier of the chemist. Okay, so all these pieces of instrument, this is a picture I have taken it from a government website. If you can see there somewhere, if you see there dbtindia.gov.in, if you can see the cursor there, dbtindia.gov.in, that means a government institute. DBT is Department of Biotechnology. This is the Indian government institute. And now you know very well, government generally provided what's your services or what's your institutes are there, they will provide their services at a subsidized rate, and those are coming at a subsidized rate. You can see there are some of those instruments, for example, this Maldi top. So 2000 per sample, 3000 rupees per sample, 6000 rupees per sample. So lots of those things are there. Private industries and profit for profit laboratories, those rates are mentioned there. So for different organizations. So try to understand that just one sample will cost you 3000. So if you have 10 samples, 30,000 are gone there. Why I'm mentioning that, if money is involved, that means these instruments are very costly. Those are sophisticated. That means yes, somebody who is handling those, or somebody who is maintaining those instruments, then that person will get nicely paid. That's what I want to mention, that yes, if you become expert in one area, then definitely that would bring niche area to you. Now some of those things again are mentioned there. For example, if you want to handle the UHPLC ultra HPLC, then 5,000 per sample. For the private organization, it will cost 7,000 rupees. So try to understand these things are very costly, and some of those instruments which I have mentioned, this is just tip of ice. This is not a comprehensive list. Let me caution you guys. This is not a comprehensive list. Okay, this is just I'm mentioning few of those which I come across, or which I have might have learned it little bit here and there. Okay, lots of instruments are there in the chemistry field, and those who is going to build it in the end, again try to understand, as an engineer you have to build it. Again, next slide will tell you lots of those things. Now one of this piece of instruments shown there, it's very simple one. It's called as combi flash, which is written there, combi flash, combi and flash. So no, sorry, not combi flash. What do you call rota vapor? Okay, combi flash is something different. Rota vapor, rota which rotates. Okay, rotates, and vapor basically is the solvent will get vaporized there, and then will get condensed, and then will accumulate there. Okay, and they are shown with the different colors so that we can see that. Basically you heat that what's your reaction mixture is there, you solvent evaporates there, gas will get condensed there, you circulate the cold water or cold ice cold water, and then condensation will happen and it will get accumulated there or collected there. Very simple instrument. If I one of the company which makes it or which is pioneer in this field, which is called as Buki, which is a German company, and if I want to purchase this one, at least it will cost me 6 lakh rupees. Very simple piece of instrument, but lots of Indian players are there, and if I want to purchase it from Indian players, they will give me 50,000 rupees. So try to understand, see the kind of a difference there, price difference there. So that's what I'm trying to challenge you guys, that if you want to make your career, definitely you should look forward to such kind of instrument where you understand the nitty-gritties of it, understand the construction and working of it, make your life there. Once you settle down there, you can start your own business there, consultancy, whatever you want to. Again, what's your instruments? Just now we have mentioned some of those instruments. For example, NMR, which comes from Bruker, where those are sitting, America. Shimadzu, from the name itself, we can say that it's a Japanese company, right? Then water, HPLC. Just now we're talking about HPLC. That water is again somewhere. The headquarters is in Massachusetts, again USA, which is one of the state in United States. Rikagu, the name itself tells you that it's a Japanese company. Headquarters is in Tokyo. Buki, just now I told you about the rota vapor. That is Switzerland, not the German, Switzerland. Okay, European country, and the head office is in Switzerland. So try to understand, all these are somewhere either sitting in Japan, advanced countries, or United States, or European countries. Somewhere, if you want to bring that business to India, if you want to make your career there, yes, definitely you have to take those steps, and those small steps, I would say, in early of your professional career. Okay, now we are switching little bit gear there. Applications in case of pharma industry, if you see there, this is a actually in pharma industry, if you understand cooking, then it's kind of a cooking. You need a vessel. Now this vessel, if you can see there, it is engineered properly, right? Because it needs lots of things. Because in case of cooking, we can take the utensil to the what you call wash basin. We can wash it there, but we cannot take this big piece of um, what do you call what's your collector there, okay, or what's your this reactor there, right? So we cannot take it. So basically water has to come here. It should have washing water, then washing. Once that is washed, it should have the drainage line. At the same time, it should have, let's say we are doing the reaction at higher temperature, then temperature should be provided, or sometime we have to cool the reaction, then hot, sorry, the cold water should be, jacket should be provided for circulation of cold water. Sometime if the heat is evolved, then we have to need something which can take off the heat. Less gas is evolved, then we have to take off that gas. Sometimes the reaction should be done under what do you call oxygen less condition. So we have to completely pack the reactor. So lots of requirements are there, and all those things, who is going to do it? Engineers are going to do it. So that we are talking about one of the formulation, which is called as tablet, not the tablet which you play with. The tablet, when we are suffering from certain diseases, we take that, we put it in the mouth, and then that is solid in nature, and then drink it with the water, right? Or push it down into the GI tract with the water. So these tablets have lots of variables there, and then let's quickly see this video, which will define lots of things there regarding this tablet, that lots of variables are there. We have to take care of it. So this is coming from one of the pharma industry. So if you can see there, the tablets basically how to punch it. Okay. So why do we have to punch it? So that the tablet should not break down when it is traveling from the industry to the medical shop, or to into the patient's hand. Till the time it is consumed, it should stay intact. So lots of things are there. But at the same time, when it goes inside, then it should disintegrate. So then that force should not be applied very vigorously, or that should not be big enough, or it should not compress it to that extent that it will not break any time. So we have to ensure lots of those things. All those things are automated nowadays. You can see there, everything is, this is a put in a piece of computer. All the commands can be given, what kind of rate you need it, output rate, input rate, compression rate. Lots of variable factors are there. All those factors can be defined onto the piece of computer. So try to understand there is a communication between this machine, what's a tablet punching machine, and computer. So there is a role for each and every one, mechanical engineer, or whether that is a computer engineer, or whether that is a ECE person. So try to understand. You might be coming for different programs here, but everyone has a role there, whether that is a field of chemistry or whether that is a field of pharma. So let's keep on moving. Now, this is another weight granulation process. Quickly, we'll look at it, how the basically tells you different kinds of um, what you call jars, and equipments are used there, and those will weight granulation process. So this is one of the process where before we do the tablet punching, okay, so granules are made. I'm not going to define what are granules, so you can look at it. Everything is automated, those things, and those happen in really wonderful fashion. Those, there should not be any mistake there, that we have to understand, and lots of those things happen in automated fashion nowadays. Very you can see different kinds of jars are there. A particular requirement is there of the pharma industry. If we can meet that, that would be really great. let's keep on moving. I guess I hope we got what we have to understand, because there are a few things which I wanted to touch base with before we lose the time. Um, Let me go to the next slide. Okay, this is quickly we'll see the tablet coating. Basically, coating is done for different purposes. Sometimes we have acid inside the stomach region when we consume it through oral route. So sometimes few compounds are sensitive to the acid, some are sensitive to the bases. So you can see quickly this one. It's not a very big video. So quickly we'll see that how that happens. So basically, it's red colored. Purposefully they have put it so that we understand that how that coating happens there. Coating, the name itself tells you that something is covered, the tablet is covered with some particles or some material, so that it will not break into the stomach, or it will not break into the intestine or something like that. Lots of those different purposes are there. So you can quickly see that how that is rotating, how that spraying is taking place, and you'll see that how that spraying is happening. Now the spraying happening of the liquid, and that should get dried. It should not stick to each other. So lots of variables are there, lots of things are there which we have to take, lots of parameters we have to consider. Now so you see that slowly that is spraying, that whatsoever things we have to coat it with. Now the change in color is happening there, and you can see that slowly that is completely getting coated with the material which we want, so that it will serve the purpose. Okay, so again we'll stop it here, and we'll keep on moving. um, Now, this is really important. This one, I guess, softwares for chemistry. Okay, so few softwares: ChemDraw, metabolism prediction, NMR prediction. NMR, just now we have seen it. Docking and simulation. So ChemDraw. So basically, it helps you to draw different structures beautifully, nice, very nice. And this ChemDraw I'm using from last 20-25 years. I see that people are using it, and still that ChemDraw is sold by Cambridge Institute in United States, and it's very costly. They sell it in dollars, and it's not affordable. So somewhere, if you can build a program which can define the structures, or which can let us draw those structures. This is a simple thing I'm mentioning. If you can see there, the bonds is going down, here the bond is coming up. So different kinds of rings are there. So this is what the ChemDraw does. Not only that one, it has immaculate number of tabs and sub tabs there, so that we can use those to our own gain, to draw structures, to draw figures, and lots of those things. Okay, so ChemDraw is for that purpose. Next one, metabolism prediction. I'll not go in detail metabolism. So basically, when you take a drug undergoes metabolism. Basically, body will try to remove that drug from our body. Okay, why it does it, we'll not talk about that, but try to understand this drug development of drug takes 10 to 15 years. Okay, and it takes about 3.5 to 4 billion dollars. I'm talking in billions, I'm talking in dollars. So if we have to reach one molecule from the chemistry laboratory to the hospital bed, then it takes 10 to 15 years. It takes 3.5 to 4 billion dollars, and though that kind of resources. And if body fails this drug, okay, in the end, then we lost those, what's your investment we have done it, of the time, of the people, and of the money. So if we can predict that with the help of computer, then there is nothing like that. Okay, that is a big help to the drug design people, so, or the drug industry. So metabolism, so they can predict it. So basically, you give the structure, give all the command. Nowadays you have AI, right, and that can help you. There are kind of like fixed set of reactions which happens on particular functional groups. You have understood those functional groups. So fixed set of reactions are there, and if we can all those things, if we can get help of the computer, then that would be really great. So lots of applications are there which predicts the metabolism reactions which will happen to a particular molecule. Basically we have to feed that structure of that molecule, and that will tell you that what are the kind of metabolism reactions those will happen inside the body. So this is metabolism prediction softwares are there. Then NMR prediction, just now we're talking about in an early part of this talk, that NMR. So basically this NMR, this is a structure, this is a NMR structure there. So this is a chemical structure there, and this is a NMR given there. So these are the peaks, different peaks you see it there. So sometimes it takes lots of time to synthesize compound in a laboratory. So if you don't want to do that, so quickly you can give it so to the predictor, this structure, and then it will predict for you. So that is really great. That is one thing. And then once we get the NMR, then we can check the actual one and the predicted one. So that helps us to understand whether this is the right molecule or not. So that's really great. So then this last piece, which is coming from the software industry, docking or simulation. What is meant by docking and simulation? Various biomolecules like proteins, carbohydrates, lipids, which are present inside our body. So those interacts with the drugs which we take it for treating a particular disease. Okay, those drugs interacts with these biomolecules. So these ribbons, whatsoever you see it, those are protein molecules. Okay, and this is a small molecule. This is a drug. Drug interacts at a particular site. Okay, it goes and meet there. An interaction happens, and energy minimization takes place. So I just now told you that in order to develop a drug, it takes around 10 to 15 years. It takes about 3.5 to $4 billion. And somewhere, if we can predict whether that molecule is going to fail or whether that molecule will be successful with the help of computer, then that would be really wonderful. That is called as CAD, CADD, computer assisted drug design. Okay, lots of softwares are available in the industry, where I'm saying industry, into the globe. Different kinds of softwares or docking softwares are available, which you can utilize to predict those interactions of the drug molecule with the target molecule or with the biological molecule. So those helps in the drug design aspect. So this is another piece of things where you can contribute as a computer scientist there. Now we are turning our attention toward the biology field. You might have seen this piece of instrument which is shown there. What is that? Which is nothing but oximeter. During this COVID time, we have realized it. Yes, lots of people suffered from this COVID infection and they lost their lives. And this oximeter, small drop in oxygen content inside your body, you can lose the life, right? So this piece of instrument tells you how much oxygen is there inside the body. This oximeter, it's very small piece of instrument. How much it costs actually? Maybe nowadays maybe 1,000 rupees or maybe a little less than that, but at that time 2,000, 3,000 or maybe more than that. But again, try to understand, Indian government, even at that time, Indian government was promoting the ingrown industry. Okay, inbuilt industry, we should have it for these medical instruments in India. Okay, homegrown, you can say. Okay, most of these small, even the small piece of instrument was coming from China. Okay, so Indian government wanted to promote such kind of industry prosperity of this instrument industry in India, and still it is promoting there. Not only this one, all the medical instrumentation facilities they want, like industry, they wanted to build it here. And even at that time also, like later on few pieces came, Indian government banned the import from China. But at the same time, people were saying that, okay, what's your instruments are available in build. They were little costly, but again, they were not manufactured in India. Basically, we were just buying the parts from China and assembling it here. Okay, so that's what it was happening with oximeter at that time. I don't know now. But yes, we can show our presence there. If you understand how that works, that would be really great. So oximeter, how much it costs? So just now we talked about maybe even 1,000 rupees. If we have a population of 1.4 billions, right? That comes to 140 crores. So even if we need some of those, even 1/10 of people or at least 100 of people, then still we can say lots of those things. Oxygen concentrator, this was another piece of instrument which we needed and desperately needed during that COVID time. Lots of people lost their life. The name itself tells you oxygen concentrated. Oxygen is needed inside the body in order to generate energy in the form of ATP. Okay, ATP is the energy currency, we utilize it. And oxygen concentrators, so the name itself tells you it concentrate the oxygen. Okay, so this piece of instrument, and at that time we didn't have enough and we were purchasing, we are getting gift from different countries, and there was havoc, if you can recall. So somewhere we can build this small industry there which can help to to grow. One thing, for themselves. Second thing, they will help directly or indirectly to the India or to the Indian government, right? Or to the people of India. This is another piece of instrument which is called as glucometer, because India is considered as a hub of diabetes, right? Glucometer, the name itself tells you it measures the glucose content of blood. Okay, and when higher glucose is there, then the person suffers from diabetes. Many of you might be suffering, or many of your near and dear one might be suffering. This is a small piece of instrument. You just put a drop of blood there onto this strip, and then it tells you what is the reading of your glucose content there, right? So very not that costly. Maybe 1,000 rupees maximum. I'm talking maybe 700 rupees if you get it at a discounted price. So maybe 700 to 1,000. Although it's not that costly, but try to understand. Just now we have mentioned population of India 140 crore, right? 10% of the people are suffering from diabetes. Try to understand how many 10%. WHO says that, I don't know at what time they have mentioned this, but more than 100 million. Just now I was reading 100 million people are suffering from diabetes in India. 100 million, 1 million is equal to 10 lakh people. So you can count that number. So enormous number. So if you are selling this glucometer, forget about the glucometer, the strips, how much that strips cost, that cost around 700, 800 or 1,000 rupees for 50 strips. Each time that strip you should be changed. Okay, for each reading you have to change that strip. What's sure that strip is shown there. Okay, so that strip cost you almost let's say 1,000 rupees divided by 50. How much it comes? 20 rupees, right? 15 to 20 rupees one strip costs you. So even if you become expert to develop that strip, if you can bring something great there, then there is no joy, and then there is no kind of like limitation there for your growth. Okay, this is another piece of instrument which we came across during the COVID time. PCR machine. PCR stands for polymerase chain reaction. Basically, that virus, that COVID virus, right? That whether we are infected or not, that will put certain things inside our body. So that virus pieces are there. We take it blood, and then we concentrate, and then but even though what's your that what's your virus is there, that is not enough. So basically, we try to make number of copies of that, and a number of copies are made through polymerase chain reaction. PCR machine. This is a machine. You will find it in every diagnostic lab or every sophisticated biology lab. So heavily used by the biologists. So if we can develop that, that would be really great. This is another piece of instrument. This instrument you might have seen, MRI scanner. Most of the good hospitals has it. I have mentioned the refurbished cost 3 crores. The newer piece of instrument will cost you almost 10 crores, and then prices vary according to the what's your variables are there, requirements are there. So MRI scanner, so that scans your brain, what is going on there. So that piece of instrument is very costly. Some of those I have listed it. There's very few, not even if you visit any biology lab, any hospital, you will see number of instruments are there. Try to again understand. Let's say this what were we talking about? We are talking about MRI scanner, right? The doctor doesn't understand how to build that MRI. Okay, he just understand how to use it. I mean, when I'm saying you how to use it, how to predict actually what's your scans it has given, how to read those scans and then make some judgment there. He doesn't know how to build that instrument. Who knows that? Engineer. Okay, again, most of the time those are attached to the PC. All these instruments are attached to the computer. There should be a communication between instrument and computer. If we understand how to build that language so that these two guys can talk, so that life becomes easier, then who is going to do that job? You guys as a computer scientist. There is a enormous need of you guys to make your career there. It's up to you guys whether you get attracted toward the chemistry and engineering phase interface, or whether you get attracted toward the biology and engineering interface or some other field. It's up to you guys. Sky's the limit for you guys. Some of those instruments I have mentioned it there. The scanning electron microscope, which you can understand in case of chemistry once you land here. A microscope, basically it enlarges the image there, right? It's the basic instrument will cost you 1.5 crore. It can go, the price can go higher depending on the requirement. It can go to 3 crore, 4 crore, depending on the story. X-ray, even that small thing, it will start from 1.5 lakh to 50 lakhs, the kind of instrument we need it in hospital. So all those things need your presence there, your expertise there, your acumen there. So try to understand limitation is from our side, not from the application side. Now, this is a simple piece of instrument. I hope all of you use it. Toothbrush, right, early morning to clean your teeth. But what is called as electronic toothbrush is available in the market, which does a really good job. It cleans. You just press that knob there. If you can see there, there is a knob there. You just press it. It vibrates and then it cleans the teeth. You put it in the mouth or against the teeth, and it cleans it. So this electronic toothbrush almost cost around, I think, 3,000 to 4,000. So when I was purchasing for the first time in United States, when I saw the price, oh my God, I thought like, okay, if I buy a simple brush, that will last for my entire life, right? But then when I started using it, there are, yes, lots of advantages are associated with this brush, that it cleans it very immaculately. Um, and what's your cleaning of teeth, it happens, then you know very well that those are not going to decay very soon. Okay, so that you have to understand. That electronic toothbrush, I just now mentioned the price. So there are not many players are there in India, and you cannot find it very easily on the medical store. You have to purchase it on Amazon or somewhere else. Um, Very rarely available, and very few buyer are there because it's costly. But somewhere if the prices come down, definitely lots of people will purchase it, knowing the importance of it. So try to understand. We have to find, and this one electronic toothbrush or what's your things are there. Lots of other things are also there. So when we are doing anything in our life, we have to keep our eyes open, whether we are in the laboratory, whether we are in the classroom, whether we are in the market, wherever we are going, we have to keep our eyes open so that we are looking for the problems, what society is suffering from. Once we know the problem, then try to find out the solution as an engineer, how you can solve that problem. And that solving the problem, it gives you satisfaction. Not only satisfaction, it comes back in the reward form, reward in the form of money. So you'll get the money. So it's up to you guys. Thank you very much for listening to my talk. I'm almost done. I do not know that last part questions. I have put it there. If there is any way you can ask me question, I'll be more than happy to answer your questions.